So I think with that, we'll try to get started. Stay here. So we're going to start our first session with Nat Rothman, and we have a session plan now that will take us through a series of opening talks to set the stage. And so, Nat, we'll let you take over here.
So I've figured out how to remove my title slide and put it back. Let's see if I can move a little bit further. As far as that is variability for sure. Variability in the quality of your speakers, I think, as far as their technical know-how. Let's see. I'm going to try other buttons and other options. And no, I'm sort of stuck here. It's sort of dichotomous. It's either on or off at one side. Can I get some help in how to move this ahead? The right-hand button. Yeah, I'm pushing that. Nothing's happening. Ah, now it is. I have to push it hard. I have to point it there. Oh, there we go. There we go. All right. Let's start. And I'm gonna have to reset my stopwatch. I've already wasted a minute, or maybe my talk has to be short by 1 minute. Anyway, well, thank you. I want to thank the organizers of the meeting, of this very interesting meeting, to give me the opportunity to talk. Today, I'm going to be talking about what do we know about individual variability and its contribution to disease. There are many sources of individual variability. Today, I'd like to focus, for the most part, but not exclusively, on genetic or inherited variability. And what this slide shows is a wide range of different types of variants, of genetic variants, starting with very small variants, single nucleotide polymorphisms, to larger variants, microsatellites, all the way to large chromosomal rearrangements. And the technology to detect those ranges from molecular genetic methods all the way to cytogenetic detection methods for these larger events. So how much variability is there in the population when we talk about inherited variants? Current estimates, these are still approximations, is that there are about 10 to 12 million common SNPs, single nucleotide polymorphisms, with more than 10% MAF, that's minor allele frequency. There may be something like 30 to 50 million uncommon SNPs with minor allele frequencies down to 1 to 10%. And it's possible there may be more than 100 million rare SNPs with minor allele frequencies down to less than 1%. There are so many variants or so much genetic variability that some people wonder if there may be variants in almost every base pair and almost every one of the 3.1 billion base pairs in the human genome. So there's a striking amount of variability. Now, the current technology that we have, before we get into deep sequencing or next generation sequencing, the current technology taking advantage of linkage disequilibrium and using off-the-shelf chip technology provided by companies such as Illumina or Affymatrix, interrogate or measure only common SNPs. So maybe 10% of all of that genetic variation for single nucleotide polymorphism. So again, just one type of genetic variant, and current technology is measuring about more than 10% or so. There's a tremendous amount of genetic variation that so far has not been analyzed. Has not been analyzed in association studies and genetic epidemiology studies that have been conducted to date. And one thing that's really striking that's emerged out of the 1000 Genome Project is that there is a striking amount, and this provides some of the data for the slide I showed you before, is that there's a striking amount of variation in this lower range here, down to under 10% and under 1% that you can see here. So again, the idea is there's an enormous amount of variation, and we've only literally just begun to start studying that. So then the question is, well, is there any evidence that the amount of variation that we've studied so far actually contributes to susceptibility for disease? So this is a slide that was updated as of, I guess, 17 days ago or so, April 1, 2012. So this focus is just on cancer, and these are all the chromosomes, and each color represents a different tumor. And if you could see the legend down here, you would see that there's been one association for liver, 10 for CLL, chronic lymphocytic leukemia. They range from some tumors where only a few variants have been found to some tumors where a striking number of variants have been found. For example, there are 48 hits or findings or loci that have been identified so far for prostate cancer. This is an extraordinary accomplishment up until the era of GWAS. If you look at decades of epidemiologic studies looking at prostate cancer, there was essentially no risk factor that had been identified for prostate cancer, with the exception of being male and being older. And that apparently applies to Warren Buffett, who was just diagnosed with prostate cancer. Fortunately, it's early, so you don't have to sell your shares in his hedge fund. But this is a ubiquitous disease, but again, whose etiology is really unlike many other tumors, where substantial inroads have been made in trying to understand the underlying etiology and identifying risk factors. This is a tumor that really had escaped the best methods for trying to understand what caused it. So within a few years, we have 48 different genetic regions that have been associated with prostate cancer. Very striking. Another thing that's very interesting about linking this enormous, or studying this enormous variability and trying to link it to susceptibility is that there are a number of regions, and these are identified by these rainbow colors here. There are a number of regions on the genome that appear to be associated with multiple tumors. This is very exciting because it suggests that these regions may hold the key to some very basic underlying biologic understanding of the factors that give rise to cancer. So these, I think, are amongst the most exciting observations. I won't yell out the different loci, except maybe this is a very interesting one on chromosome 5, and the terching, which is involved with regulation of telomerase, and TP63 is a tumor suppressor gene. So again, these are talking about inherited variants, sources of variability that seem to play an important role in susceptibility for disease, in this instance, cancer. If I had a similar slide for diabetes, for coronary artery disease, you would see many findings as well. If I had a slide for obesity, for example, there are actually dozens and dozens of findings that have been associated with obesity. So there is a striking amount of genetic variation. We have only begun to study to what extent and these variants are associated with susceptibility to disease, and there is every expectation that there will be an explosion in the number of new genetic findings as the technology emerges to allow us to interrogate the component of the genome where lower allele frequencies exist, below 10% and below 1%. Now, what I'd like now to do is to talk about how information and knowledge about variability can be incorporated into studies where we're looking at relationships between environmental exposures defined in various ways and disease. What's the rationale for doing this? Well, the rationale, as we just heard before, is manyfold, but some that I'd like to show examples of is that it can help us obtain mechanistic insight, it can clarify dose-response relationships, and more effectively evaluate low-level risks. It may identify new environmental health hazards, and ultimately, and hopefully, develop more effective prevention, screening, and treatment strategies. And what I'd like to do is to provide several vignettes from researching one tumor, bladder cancer, which is a wonderful model for studying gene-environment interactions for several reasons. It also is the sixth most common tumor, and it is one of the most expensive tumors from a public health perspective, since once someone is diagnosed with bladder cancer, since most patients present with early disease, they need to be screened again and again and again to look for recurrence. So it actually has a fairly high public health burden on the population. And what I'd like to do is talk about one study in particular and show a few examples of how we've looked at susceptibility, looked at variability, to identify what we think are some susceptibility sites, and then look at how that interacts with some important exposures that play a key role in bladder carcinogenesis. And I'll talk about mostly genetic variants, but I'll also talk about some other sources of susceptibility, since there are a wide number of various endogenous sources of susceptibility. Some are genetically mediated in total, some have a modest genetic contribution, and some are independent, we think, of any genetic component to them. So this is a study we carried out in 5 parts of Spain. We conducted the study in Spain because bladder cancer rates are relatively high there compared to other parts of Europe. We were particularly interested in environmental and occupational exposures. We put a lot of effort in collecting exposure data, including complete the residential histories, looking at fluid intake, urine pH, urine frequency, so that we could try to put together a model of bladder carcinogenesis, looking at individual susceptibility and variability, and then look at how that interacts with known or suspected bladder carcinogens. So one of the first exposures, one of the key exposures that was a priority in this study, was to look at disinfection byproducts, their potential interaction with certain metabolic polymorphisms and risk of bladder cancer. Now, many of you are probably familiar with disinfection byproducts, but the gist is they are ubiquitous in all water supplies, disinfected with chlorine. We can measure them in the water that we'll be drinking today coming out of the Potomac. The precursor levels are much higher in surface than groundwater sources, creating a major differential exposure. They're a complex mixture of halogenated organics with mutagenic properties, and one class is called trihalomethanes. So the gist is that we chlorinate our water, which is good. We get rid of things like E. coli and other things, and that's had a major public health impact. But one of the adverse consequences of this act is that if there's an organic material present, that organic material gets chlorinated and turns out to be highly mutagenic, and we and others groups have found data that suggests that this is actually associated with bladder cancer, possibly rectal cancer as well. So a tremendous amount of effort in this study was made to attempt to estimate people's exposure to disinfection byproducts. Lifetime residential and water source history was gathered from each subject, and ultimately long-term average cumulative peak, et cetera, exposures were calculated. Now, the a priori hypothesis in this particular study was to look at the interactions with intake of these chlorinated compounds and variants in 2 key phase 2 metabolic genes, GSTT1 and GSTZ1. Now, interestingly, GSTT1 activates these compounds. GSTZ1 clears these compounds, and their functional variants in both of them. So this shows the initial report that we had with a dose-response relationship between trihalomethanes. This is concentration per liter in the x-axis, and this is risk and odds ratios. And you can see a modest dose-response relationship, somewhat significant trend, with risk going up to 1.8. So then we stratify the effect by GSTT1 genotype, in this instance the null genotype versus wild type. And what we see, sort of interestingly, is that dose-response relationship is completely flat in this group and is somewhat sharper here, with risk going up as high as 2.2. Now we stratify by the second functional polymorphism. Again, we see a flat dose-response relationship, but an even sharper dose-response relationship here amongst the group that a priori we thought would be susceptible, with risk going up to 2.9. Now, if we combine these two together, and we now look at people who are exposed to high levels of trihalomethane, who are unfortunate enough to have both variants of the at-risk allele, again, we see here in the population that's most protected, no evidence of an association with risk, and we see this very, very sharp dose-response relationship with the risk going up to 5.9. So what we've done is we've basically taken the population, where initially we saw an overall risk of 1.8, and we've separated them into a subgroup where there's no risk, and a subgroup who, for the most highly exposed, have risks up to 5.9. So a number of ways of looking at this. We've identified about a 6-fold variation in risk, depending upon your genetic makeup, or if you ignored risk and you simply did the overall effect, you would see a risk of 1.8, and when we separate that out, it goes up to 5.9 in one group and 1 in the other group. So I think this is just the beginning of at least providing some evidence that these overall associations that we see between environmental exposures or between drug effects and adverse effects really represent a very heterogeneous observation, and that there are truly subgroups who are at little risk and subgroups who are at high risk. And again, not to be redundant or overly cite the song "We've Only Just Begun," but we truly have only just begun to identify subgroups of people who are at the highest risk. And we expect that this kind of splaying out or differentiation of risk will become more and more extreme as we sort of delve further into genetic differences. The next exposure that I wanted to talk about is tobacco. Tobacco is the strongest risk factor, at least from a general population perspective, in bladder cancer. Though the highest risk for bladder cancer come from the occupational setting with exposure to aromatic amines. Probably 2/3 or more of bladder cancer is driven by tobacco risk. So another hypothesis of this study that we conducted in Spain was that people who smoke would be at higher risk depending upon a number of endogenous susceptibility factors. And there were 3 we were interested in looking at. Detoxification of aromatic amines by NAT2, which is N-acetyltransferase 2, acidic urine, and decreased urine frequency. So this is the only chemical slide that I'll show you, a metabolic slide I'll show you, but I just want to take a minute to take you through this. This is, for example, 4-aminobiphenyl, which is one of the most toxic carcinogenic compounds within tobacco with regard to the bladder. This gene, NAT2, acetylates that NH group, and once that's acetylated, it becomes non-reactive. So this is a protective reaction. About 50% of the Caucasian population carry variants that result in homozygous no function. So half the population cannot carry out this reaction, in which case these compounds aren't detoxified and they're free to undergo subsequent activation. They undergo activation by a number of enzymes, including CYP1A1. Then they can be conjugated by the UGT family that produces glucuronides. These are very water soluble. It enhances the secretion of these compounds, transported from the blood into the bladder, where an interesting thing happens. Many of the most carcinogenic glucuronide conjugates are acid labeled, meaning in acidic conditions they deconjugate, they sort of fall apart, and now these are available for activation in the bladder and formation of DNA adducts. So what the body has done is it's developed the system to enhance the excretion of a compound that it perceives as a toxin, goes into the bladder, but if you're unfortunate enough to have acidic urine, that becomes cleaved, that's now available to form DNA adducts. So we have a delivery system of a carcinogen into the bladder. On the other hand, if you have a tendency to excrete frequently, this is good because you're getting rid of the stuff. So ultimately, our hypothesis was that if you were a slow acetylator and you had an acidic urine and you had a tendency not to void frequently, particularly at night, that this would increase your risk. So this shows the risk for each of these 3 risk factors, and we indeed found significant increased risk for being a slow acetylator, having acidic urine, and not voiding at night. So here the referent is voiding 2 or more times. So what this means is that at nighttime, when one has the urge to void, get up and go. This is good. Get rid of it. And in fact, overnight, what's happening is all these carcinogens that you may, even this observation was present even among non-smokers. All of these carcinogens that you're exposed to, they're being metabolized and delivered into the urine, and you're sleeping, and it's just sitting in your urine. Urine also has a tendency to get more concentrated overnight. There's some resorption of water, so it's just more and more concentrated, more time it has contact with the urothelial cells. This we found was sort of striking, a clear tendency. You void more, you're protected. And again, in smokers and non-smokers. So all 3 risk factors were associated with increased risk of bladder cancer, as we had hypothesized. And then we asked the question, let's look at the overall effect of smoking. And then let's separate it out, stratify it by these risk factors. So in this particular population where they use black as well as blonde tobacco, very potent, it's full of aromatic amines in Spain and some other parts of southern Europe, there was a very high risk, a risk of 7.5, an odds ratio of 7.5 for smoking. Now, if you were a smoker and you were unfortunate enough to be a slow acetylator, your risk went up to 9.7 versus 4.9. And if you were a smoker and you had this characteristic, as well as having a tendency not to void at night and having acidic urine, which it turns out is a function of your body surface area and diet, that risk splays out into a risk of 16.6 and 3.1. So about a 5-fold difference. So here, again, with this notion of we take a risk factor, we start looking at internal endogenous sources of variability. Some genetic in this instance, some non-genetic sources of endogenous variability, which are very strong and in some instances can be as important as genetic factors. And we see, again, this splaying out of risk of about a 5-fold difference. So that 7.5 became a 16.6 versus a 3.1. So now I want to move into the modern age. This initial work is what we call, people who do genetic work call candidate gene work, where we came up with some genes. Not that we were particularly smart. Many people had thought about these before as being sources of variability that might be susceptibility genes. And so they were a priori genes, so we measured them specifically up front, and we had the good fortune to show positive results in the expected direction. Now let's move to the new age of genomics, where we take an agnostic approach, because in general, this candidate gene approach has not been very successful for most diseases, in fact, for almost any disease. Most candidate gene associations that have been observed over the last 15 years or so ended up being false positives, and there are a number of reasons why that ended up being the case, but that gave rise to a sense of nihilism about the candidate gene approach. But fortunately, around the time people realized that almost everything that was published was wrong, an agnostic approach became enabled through the design of advanced technology and chips that allowed us to carry out genome-wide association studies. So this was a study that we carried out, had several phases of bladder cancer, ultimately included 12,000 cases and 53,000 controls from 20 studies. And this shows previous GWASs had identified associations at these loci. Our study, there it is. Our study identified additional loci, and then in a collaboration that we had with MD Anderson, we identified additional loci. Now, we then asked the question, we said, okay, well, this is interesting. We've identified in the aggregate across all the groups that have been conducting GWAS, we've identified 12 variants, 12 loci that are associated with bladder cancer. And then we asked the question, well, let's look at the risk of smoking and look for the presence of interactions with each of these hits. And we found that looking at additive interactions, we found that 7 of the 12 SNPs showed evidence of this type of interaction. So that we thought was pretty interesting. And then we thought, well, how can we put this into a public health perspective? So we carried out a number of calculations, and we're lucky that we had a lot of population-based data to help us with this. And this is the question that we asked. Let's look at the cumulative 30-year absolute risk for bladder cancer in a 50-year-old male in the United States. And then let's look at that risk by smoking, and then let's look at it by genetic subgroups and see what that tells us. So if you first look here on the left, this is a cumulative 30-year absolute risk. So this is not relative risk. This is basically saying a 50-year-old male in the United States over 30 years, if they're a non-smoker, will have a little bit more, will have a 1.3% chance of developing bladder cancer, absolute risk. Now, if we look at former smokers, it's 3%. If we look at current smokers, it's 6.2%. So that's what you see overall if you ignore genetic factors. So then we created a model using all of these genetic risk factors and came up with gene scores, basically putting people in quartiles of low versus high genetic risk. So this is a gene score 0, 1, 2, and 3, going from low to high risk. So if we look at the risk in the group that had the least genetic risk, you see that instead of having a risk of 1.3, never smokers have a risk of 0.9%. Former smokers have a risk of 1.4% instead of 3%, and current smokers have a risk of 6.2%, drops down to 2.9%. So now let's look at the risk difference. All right, the risk difference here is defined as the difference between the current smokers and the never smokers within this gene score. So there's a 2% difference. All right, the current smokers have an absolute risk of 2.9%. Never smokers have an absolute risk of 0.9%. So 2.9% minus 0.9%, 2%. Now let's go to the high genetic risk score. Risk of 1.7% in never smokers, all the way up to this huge risk of 9.9%. That means Again, a 15-year-old white male living in the United States who smokes has almost a 10%, a 1 in 10% chance of developing bladder cancer over the next 10 years versus a 6.2% change. So what you see, again, the notion, we're now taking absolute risk and we're separating them. So for current smokers, if you have low genetic risk, your risk is 2.9%. If you have high genetic risk, your risk goes up all the way to 9.9%. What's the risk difference here? The risk difference is 8.2%. It's a lot bigger at the high end. The test for additive interaction was highly statistically significant. And again, that risk difference is the difference between the absolute risk for the current smokers versus the never smokers. So what are the public health consequences of this? Well, we would not intend or imply that we should selectively counsel people with this in the high genetic risk category. And again, this is just preliminary data. It needs to be replicated, et cetera. We would not counsel only people with high genetic risk to stop smoking. We would not screen only people with high genetic risk to stop smoking. But these calculations allow us to make this statement that within these data and within the accuracy of these estimates, and this is just a first effort, we would say that the impact of eliminating smoking in 100,000 current smokers in the highest versus lowest genetically susceptible subgroups is this. If we could eliminate current smoking in 100,000 people who had the highest genetic risk, we would eliminate 8,000 cases. If we did the same in the subgroup of people, 100,000 people with low genetic risk, we would eliminate only 2,000 cases. So this begins to give you a sense, if our calculations are true, this begins to give you a sense of the potential application that could be very, very powerful of looking at risks, at environmental risks, at looking at drug risks, separated out by genetic categories, if these are true positive findings, if these models ultimately replicate. And that may have important public health implications as far as counseling, as far as screening, and at the same time, we would not say, let's go ahead and start screening or selectively counseling people. That would be ridiculous. Also, bladder cancer is only one of the many diseases associated with smoking, and one really needs to do, and this is being done now, look at these effects in cohort studies where we look at the impact of smoking on all different diseases, and then we develop genetic models that allow us to really say, if you're smoking and you have this genetic makeup, this is what we estimate to be your absolute risk. If you have this for this disease, for this disease, for this disease. If one were to integrate across all diseases, and one could say, with this combination of genetic factors, you are at substantially higher risk for a variety of different diseases. And another population might be at a much, much lower risk. This has implications, I think, as far as environmental risk assessment, occupational risk assessment, and again, looking at risks from other types of substances and exposures as well. So again, this is just a vignette, but it's an example, and I believe it's one of the first examples where we've been able to take what's beginning to come out of GWAS studies and more extensive genetic analyses will follow, integrate that with exposures, and start thinking about the potential implications and applications of these genetic differences. Because these differences are non-trivial. 8,000 versus 2,000 cases, that's a pretty big difference. And again, I've said it before, we truly are not advocating, and in the workplace, which is where I come from, as far as my main area of research is in the occupational setting, we would never ever advocate screening workers beforehand and only allowing the so-called non-susceptibles to work in a given workplace. But these kinds of differentiation in risk given certain types of exposure have always been hypothesized to exist. We and others are now contributing data showing they, in fact, do exist, and there'll be a lot more evidence in the future. How to deal with this is a major issue. But I think what compels me is that there now is evidence that these are real phenomena. We have hypothesized about these literally for decades, and the reality of these associations and these types of interactions are now upon us, and we have to think about how best to use them. So ultimately, in the context of bladder cancer, what we're doing is developing a model where we can look at all the various exposures, occupational, environmental, tobacco, and look at all the different genetic factors and endogenous factors as well, like having a low urine pH, voiding frequency, as well as variants in genes and loci that whose function are unclear at this point. Some of them are linked to tumor suppressor genes. One of them is linked to this very important locus with TERT. And the idea is to try to put together a model where we understand how these various types of variability end up being susceptibility factors and how they react and interact with the variety of exposures that drive bladder cancer risk. So in conclusion, I think it's exciting and very important to integrate knowledge of human variability into studies of exposure and disease. There are a lot of opportunities to do this, and there are many rationale for doing this. Again, just to repeat, it can help us obtain mechanistic insight, clarify dose-response relationships, identify new environmental hazards, and hopefully develop more effective prevention, screening, and treatment strategies. And I'd like to just close with a comment from President Woodrow Wilson. President Woodrow Wilson, I learned, was a major supporter of the National Academy of Sciences and actually signed the executive order creating the National Research Council. And he said, advances will be accelerated by collective intelligence, and I not only use all of the brains I have, but all that I can borrow. And I thought that was very fitting, the challenge to understand all the different sources of variability, to understand how that plays into susceptibility for disease, and to understand how that interacts with the exposures that many of us here are driven by to understand and to control. Requires a major effort. There's a tremendous amount of work to be done, but again, I believe it is real, it exists, and it will be a great challenge that will require many people from many disciplines to figure out how to use this information and thoughtfully apply it. Thank you.
About 5 minutes for just a couple of questions from that. If anybody would like to start, just use the microphone. There are microphones on the sides.
Duncan Thomas, University of Southern California. A very nice talk, Nat.
Thank you.
You made an intriguing comment right near the end about not wanting to use genetic information to screen workers, a philosophy I think most of us would agree with. But how then should we be using genetic information for regulatory purposes? Obviously, it would be very nice to get rid of all occupational carcinogens. But I'd just like to hear your thoughts about where to go next.
Sure. I think one of there are many reasons, and I won't go into them. It'd be like bringing coals to Newcastle in this audience as far as why we would not want to screen workers and only allow the so-called non-susceptibles to work in the workplace. But many of you may be aware there are egregious examples in the past where there were genetic risk factors. What I should say is sources of variability that were claimed to be sources of susceptibility ended up not being the case, but that were distributed differently in various ethnic groups, and it resulted in discrimination. So not only was the discrimination was terrible to begin with, the science was wrong. So it was two things that were terrible, with the discrimination being the worst, but it was based upon incorrect assumptions about these variants being susceptibility factors. I think it's very important to separate the two in mind. The notion of there is susceptibility, there's variation, and that may or may not lead to susceptibility. And people can incorrectly infer and have in the past, and that had terrible consequences. So how to apply it? One way I think of applying it is that we have some carcinogens, occupational, environmental, whose carcinogenicity actually is equivocal. It's not certain. And what I believe we have a few examples of, and we will see more in the future, is that these sort of murky dose-response relationships, like the first slide I show with disinfection byproducts, will actually become clearer, showing stronger risks. That very strong dose-response relationship with the risk going up to 5.9, with a highly significant trend in the subgroup of people who were unfortunate enough to have the two variants that result in more enhanced activation and less excretion of trihalomethanes. I think that's an example where if you weren't too sure about the carcinogenicity of trihalomethanes, and you saw this dose-response relationship, this plateauing at the top, the p-value, it's significant, but it's 0.03, the risk only goes up to 1.8, and you say, here's a subgroup, and this group, I'm convinced I have really good biologic data that supports that this subgroup really biologically should be at increased risk, and lo and behold, they are. to me, and I think to others, that adds to the weight of evidence that, in fact, that compound is a carcinogen. So one, for using risk assessment nomenclature, for risk characterization, I think that we will have an ability, even in the world and all the misclassification of exposure assessment that exists, even with our best efforts, that the genetics are a tool to allow us to potentially identify subgroups and increase risk where the clear dose-response relationship will add to the weight of evidence that this, in fact, is a carcinogen. So for risk characterization. The other issue, I think, is in looking at dose-response relationships. As you said, is a tenfold factor adequate? You can see now with I consider these results very, very preliminary against the backdrop of all the work we're going to be doing with genetics. But the differences that we saw were threefold, fourfold, fivefold. So with not a whole lot of work, you start wondering about this tenfold standard factor and how good that is, because I think very soon we'll see more than a tenfold difference. So I think a second application is in the context of what levels are safe. I have done a lot of research in benzene. The current standard is 1 part per million based upon OSHA, but NIAS says it should be down to 0.1 ppm. We've done some work showing evidence of susceptibility for intermediate endpoints, looking at things like hematotoxicity, where we clearly see subgroups who have substantially greater risk, even at lower levels. Steve Rappaport is here, and he's shown evidence of effects at very low levels of exposure. If you look at the weight of evidence, I think we see that we see subgroups that will be at higher risk at low levels of exposure, and I think that can also contribute to standard setting. That we shouldn't be lumping everybody together. So those are two examples, I think, of the application of this work in the occupational setting without sort of the standard clinical genetics approach would be, well, just remove these people from the workplace. I think from a regulatory public health perspective, we have, a broader perspective where we think about the whole population. And again, the notion that, OSHA's mandate is to make the workplace safe for everybody, not just for the least susceptible.
Thanks. We have 4 people back there, and I think we'll just have to keep it to relatively short questions and relatively short answers.
Short answer is probably the key factor.
Sure. Melissa Perry, George Washington University here in Washington. Thanks for a very thorough and clear presentation. I'm wondering if we could go back to the slide just a couple back, presenting the 30-year cumulative risk. Yes, right there. So I'm sure that your group worked long and hard to come up with the gene score. Can you tell us a little bit about that, reflect on it without going into the nitty-gritty details? How valid and reflective do you think it is? How can we repeat it?
It's relatively straightforward. I think there are, and I'd be happy to share the methods with you. The paper just got rejected from a high-impact journal with not the kindest reviews, but we're now trying to, for one level down maybe, but I'd be happy to share the methods with you. The gist is that some people have come up with a gene score where they just add up the risk factors. This is weighted. So it's weighted based upon the relative risks observed with each one, and it's modeled, and then we create these quartiles. So we think it has more validity because of the fact that it's weighted by risk instead of just simply saying 1, 2, 3, 4 or more alleles. But again, happy to share with you the actual methods.
Working with this method, then you were satisfied with the results, and you want to keep working.
Yeah,
with it.
we are. We think it worked nicely. This is sort of a relatively new approach, and I want to give credit for, I didn't show my last slide, and I'd like to have that up there if I could, of all the people who've worked on this project. But Nilanjan Chatterjee, who is chief of the biostatistics branch in the Division of Cancer Epidemiology and Genetics, and Monse Garcia Closas did a lot of work in developing and applying these methods.
I was sure of it, and I was sure that your group had worked long and hard and carefully considered the system, and so I didn't want to lose that detail.
Nick?
In locales which use ozone as a disinfectant, you'd expect then, other things being equal, to have a significantly lower incidence of bladder cancer. Do you?
That's a good question. We have not looked at that. That's a good idea. It depends on, obviously how extensive it's being used, how large the population is, but that would be a natural experiment to do a comparison. Thank you for the idea.
Hi, Joel Schwartz from Harvard. So you just mentioned that stratifying by the genetic risk can help you better identify the environmental risk, and I think that's true, but I'd like to suggest that the opposite is also true. In your first example, you mentioned 2 genes polymorphisms that modified the risk of, halogenated organics that didn't show up in your GWAS study.
Good.
But the GWAS study, of course, didn't have exposure to trihalomethanes, and they may only show up in people with the higher exposure. And so we also can sometimes miss polymorphisms that may be important by not having the exposures in our GWAS study.
A critically important point, and a very short answer is, there are some studies, GWAS studies, that have just, the cases come from a case series and the controls come from another population and there's no exposure data, and that's actually good enough to look at the main effect of genes. The studies that I work with and my collaborators are more come from the traditional epidemiologic design with a big focus on exposure. And there are statistical methods, and again, I want to give credit to Nilanjan Chatterjee, who's also worked with Stephen Chanik, who directs our genomic facility at NCI, statistical methods that allow the simultaneous evaluation of subgroups based upon exposure in original GWAS data. So in fact, we now, what I showed you was the interaction of the hits identified as main effects, just looking at everybody. What we're now doing literally are running models where we take that GWAS data and we stratify by the exposure data because, you're right, these were not, it was a good observation, these genes did not show up, they didn't achieve the threshold of genome-wide significance of 5 times 10 to the minus 8. Instead, they just had very, very weak effects. Why? Because they manifest themselves only in a subgroup of the population. So you need that data, and we and other groups are now working with the data on our models to try to do that in an efficient way.
Terrific.
One last question.
Hi, my name is Matt Gribble. I'm a student at Johns Hopkins, and I'm doing my thesis on genetic susceptibility to arsenic and genetic determinants of arsenic metabolism. And those are kind of distinct ideas. And I was wondering, your talk was very interesting, but focused on risk of specific diseases. How do you feel that gene environment knowledge can help us understand mechanistic aspects of the disease process, and do you think that has implications for policy downstream too?
Right. I think that first, by taking the main hits, meaning the main effects, the overall associations that we see from things like a genome-wide scan, and crossing them with exposures, if we see evidence of interaction, that already begins to say, geez, here's a variant in a tumor suppressor gene or in a telomerase gene, and that this specifically seems to interact with this exposure. That begins to suggest a mechanism right there. Secondly, a more agnostic search for interactions, like I was describing before, where we look at interactions, basically look at interactions with all 750,000 SNPs. Well, also we expect to identify variants, and if those are real and they're robust and they persist and replicate, we think that will even further identify interactions where one understands what that gene is. Will lead to potentially opening up the door to a whole new lines of mechanistic research.
Thank you, Nat. Great start.